Good morning, students. How are you all? So, before going to start today's topic, I have a beautiful quote for you. The art and science of asking question is the basis of all the knowledge. So, simply keep on asking the question. The way you design your question, the way you ask your questions, that is really going to benefit you. So today's topic is sexual reproduction in flowering plant. This is chapter 2, unit 1, class 12th biology. And we know that flowers are the most fascinating parts of a plant. We all feel so happy when we watch different kind of flowers blooming in our garden, in school garden, in our near our house, or anywhere. So do really flowers are meant for giving us happiness, or they are serving some other purpose also? Yes, definitely they are serving very important function because they are the reproductive part of a plant. So you know the part of a flower, there are male reproductive part and female reproductive part. The plant which either contain stamen, either contain pistil, is called as unisexual, and the flower which you can see here in front, which is containing both, it is called as, we can simply say, this is bisexual flower. So flower is nothing but it is modified shoot developed from floral primordia. Primordia develop first into floral bud and then into a flower. This is the reproductive part of a flower plant, and androecium is the male reproductive part, gynoecium is the female reproductive part. Then I am talking about stamen. It is male reproductive unit. It is consisting of anther, which contain lot of pollen grains, and filament is a structure through which it is attached with the base. Filament is attached to the thalamus or the petal. And when we just observe the transverse section of a anther, we will find it there are different kind of a lobes, there are different kind of a pollen sacs, and in pollen sacs you will find thousands of pollen grains. When you just dust a flower, a mature flower, you dust it on a white paper, you will see the yellow colored dust which is fallen on your paper, which is nothing but they are pollen grains. So when I am talking about structure of this anther, this is bilobed and diathecous. You see, it is bilobed and diathecous. A longitudinal groove separate the theca, where there is a wall which is called theca. And in the cross section of anther, tetragonal structure, you see they are called as microsporangia. Inside these microsporangia, microspores will develop, or we can say the pollen grains will develop. Then structure of anther. The microsporangium is a circular and surrounded by 4 layers. These are epidermis, endothecium, middle layer and tapetum. Outermost layer protect the pollen and help in desiccance of anther to release the pollen. It will rupture when the pollen grains will release from that, when the anther will become mature. Tapetum is a very important wall which is directly in contact with the pollen grain. It nourishes the pollen grain. Whether the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissues occupies the center of each microsporangium. And when we observe the structure of a pollen grain, it is also made up of 2 layers. Outer layer, XII, inner layer is intine. There are small pores through which the pollen tubes will actually develop, and these pores are also very important. The outer layer is very thick, and that is made up of sporopollenin, and inner layer is called intine. Then microsporogenesis, how these pollen grains will form. The process of formation of the microspores from the pollen mother cells through meiosis is called as microsporogenesis. The cell of sporogenous tissues undergoes meiosis to form microspore tetad, a cluster of 4 cells. As each cell of the sporogenous tissues has potential to form the tetad, so each cell is a microspore mother cell, that is called as pollen mother cell. On maturation and dehydration of anther, the spores dissociate and develop into pollen grains. Pollen grain release with the desiccance of anther. It will rupture and it will release the pollen grain. Then pollen grain is spherical in shape, I have already mentioned. It has hard XII, I have told you, and inner layer is called intine. XII is made up of sporopollenin, I have already mentioned, which is resistant to organic matter, withstand the high temperature, acid, alkalis and enzymes also. Intine is thin and continuous layer, and it has outer layer has small apertures, I have already told you, which where germ pores, they are called germ pore, where sporopollenin is absent. Then pollen grains Cytoplasm is surrounded by plasma membrane. Mature pollen grain has two cells. One is vegetative cell, another is generative. Vegetative cell is bigger. Generative cell is small, spindle shaped. In 60% of the species, the pollen grains are shed in two-celled stage, where in other 40% species, it's celled in three-cell stage. Yani, that generative cell actually is divided into male gametes. Embryo sac. This is the female reproductive part. And when we observe the female reproductive part, we say there is a one part which is called callosal end, micropilar end, and it is also made up of some layer. It is surrounded by layer. They are called as integuments. And at some portion, these integuments are missing, which is called micropilar end, through which generally the pollen tubes will enter. And it is also 8-cell stage. Out of those, we have 2 synergid, 1 egg cell. In the center, you will see the 2 polar nuclei, and at the callosal end, you will see 3 antipodal. I will explain the function of all these 8-cell structure, 8-celled embryo sac. So pistil is the female reproductive part of a plant, we know that. And when I'm talking about ovule, arises as a primordium on the placenta. The short stalk which attaches the ovule with the placenta is funicle. The primordium grows into a mass of cell. It forms nucellus. I have already mentioned it is covered with integuments, and at one portion where integument is missing is micropyle. Ovule. Basal part of ovule is called callosal. Cells of nucellus are rich in reserve food. A single embryo sac of female gametophyte located in the nucellus, which is developed into megaspore. Initially we have talked about microspore mother cell. Here we will talk about megaspore mother cell. The formation of the pollen grain is microsporogenesis. Here we will say megasporogenesis. So I have already mentioned this. 3 antipodals, 2 nucellus, and when the pollen tubes, after the pollination, this pollen tube will germinate from the stigma. It dissolves the wall of stigma. It goes to the style with the help of a special movement, and which is called as gametropism. Megasporogenesis and development of female gametophyte. Then the nucellus region, the micropilar region, is differentiated into megaspore mother cell. This megaspore mother cell undergoes meiosis. It forms 4 haploid cells called megaspore tetad. 3 megaspores out of them will degenerate and 1 megaspore become functional, and it will divide and redivide, and it will form 8 nucleate stage. Megasporogenesis and development of female gametophyte. Functional megaspore is the first cell of female gametophyte. Its nucleus undergoes mitosis. 2 nuclei move to the opposite pole and to form 2 nucleate embryo sac, 2 successive mitotic division, and resulting in the formation of 8 nucleate embryo sac. Out of them, 3 I have told you, 2 of them are synergid. 1 is egg cell. At the center, 2 polar nuclei and 3 antipodals. Simultaneous maturation of anther and pistil. Anther and stigma lies close to each other. Clistogamous flower, oxalis and Camellia viola, they are responsible for autogamy. This means self-pollination. Xenogamy, unisexual flower, dioecious plant, pollen release and stigma receptivity is not synchronized. This means pollen grains and stigma maturations is different. Pollen either release earlier and stigma will mature later on. So it will promote the cross-pollination, and that is very important, cross-pollination. For the releasing the variation, for creating the variation, and variation are important for the survival of the species, you know that. Then we talk about different agents or agencies of pollination. So when we talk about wind pollination, there are some flowers which are pollinated through wind. There are some plants which pollinated through water, hydrophily. There are some plants which are pollinated through birds, ornithophily. Insects, anemophily. Then what is pollen pistil interaction? You know that different kind of pollen grain fall on the stigma, but generally the pollen grain of the same species will germinate. So this is called as pollen pistil interaction, and this is called recognition of compatible pollen. It is the interaction between chemical components of the pollen and those of stigma. Germination of the pollen and development of the male gametophyte. I have already mentioned this. This is a very important question when I am talking about double fertilization in flowering plants. So what will happen? When the pollen grain fall on the stigma, they get some nourishment. They develop the pollen tube. And there is a very simple question, why this pollen tube is not going towards air, toward the sky? Why it is moving toward ovule? So there is a very simple question. There are some sugar, there are some boron, there are some inositol complex which is there in the female reproductive part of a plant which is pulling them toward the ovules. So two male gametes from the pollen tubes into the cytoplasm of the synergid. Synergid guide the entry of the pollen tube. It is already containing two male gametes. One of the male gamete fuses with the egg cell. This is called syngamy, form the zygote. And another male gamete fuses with two polar nuclei, and it is called as triple fusion. It forms PEN, primary endosperm nucleus. As syngamy and triple fusion occurs in the embryo sac, the phenomena is called as double fertilization. So fertilization in plants is called as double fertilization. So you know the process why it is called double fertilization. Then we have post fertilization event. So endosperm, it is the development precedes the embryo development. There are three methods of embryo development: nuclear, cellular and halobial. The nuclear type PEN divides mitotically without cytokinesis, and endosperm is free nuclear. Then cell wall formations start from the periphery to the endosperm. It provides food to the developing embryo. If endosperm is completely utilized by embryo, then they are called as non-albuminous. If it is present, it is called albuminous seeds. Embryo development. It starts after certain embryo formation. Zygote divide mitotically, form pro-embryo, then it divide into globular and heart-shaped embryo, then horseshoe-shaped mature embryo, and with one or two cotyledon. If one cotyledon, monocot. Two cotyledon, dicot. Apomixis. Seeds are formed without fertilization in some plants. It may be developed if the diploid egg develop into embryo without fertilization. If cell of nucellus may develop into embryo and pushed into the embryo. Special reproduction is polyembryony. If more than one egg may form in the embryo sac, if more than one embryo sac in the ovule, other cells like synergid or nucellus also develop into embryo, like orange, lemon, mango, onion, groundnut, etc. So I hope you have understood this whole chapter, and this is a very simple thing what you have to learn. The main questions of this chapter is, what are the different parts of a flower? Then what are the male and female reproductive part of a plant? Transverse section of a anther. This is very important question. Many times it has been asked in board examination to label this diagram or draw this diagram and explain the process of microsporogenesis. And similarly, the female reproductive part of a plant, and that is megasporogenesis, is also very important question. And of course, then pollination, types of pollination: autogamy, xenogamy, self-pollination, cross-pollination, then double fertilization, these and development of the embryo. These all are the main important part which are generally asked in examinations. So I hope you have understood this, and at last again I would tell you the thought which I have written here. The art and science of asking the question is the basis or the sources of all knowledge.